Hey guys, this is Mr. Wayt, and this is your assignment for August 30th, Tuesday, 2022. First of all, go ahead and start with your AP Biology journal question. This is found on AP Classroom, BRNR 1.5.2, Structure and Function of Proteins. The first one is a multiple choice quiz question. The next one is an FRQ. Now you'll notice this is a little bit different. I've designed it to show you the correct answers after you respond. I know that works well for the multiple choice. I'm hoping it works just as well for the FRQ. If not, we can talk about it when I return. If you would, go ahead and pause the video at this point if you haven't gotten this far already, and then take the time to answer these questions. All right, I hope you've got to answer those questions as well as go over the answers. So let's go ahead and look at 1.5.2, structure and function of proteins really quickly. If I go up here, the first thing it wants to talk about is amino acids have directionality. Remember, nucleic acids have a directionality, 3 to 5, 5 to 3, anti-parallel. Now we're looking at proteins, and these are amino acids are the monomers of proteins. They're the most basic unit. And here's what you should know. Just like the nucleic acids had a phosphate sugar base, amino acids have an amino group, a carboxyl group, and an R group. And it is this R group that allows it to have its special properties, whether it's acidic, it's basic, and a variety of other properties. But also don't forget this, when you have a carboxyl group on the left combined with an amine group on the right, this H hydroxide and the H will make water. Water will come out of here, you dehydrate it, and then if you synthesize the rest of these two molecules together, you get your dehydration synthesis. It says there are 4 elements of the protein structure. Guys, let's just go over here and look at this really quickly. It talks about how here is your primary structure. Primary structure is just a sequence of amino acids connected by peptide bonds. We've talked about hydrogen bonds. When we're connecting amino acids, the amine group to the carboxyl group, that is officially called a peptide bond. Now, if you look at the next part over here, the green curly Q, there's a couple different names for this. You could call it a pleated sheet that kind of goes back and forth, or you could call this a helix. Sometimes they refer to it as an alpha helix, how it kind of makes this loop-de-loop like this. This would be our secondary structure. And then when we fold it back on itself, we'll start to get a tertiary structure. What should you know about the secondary structure? Go backwards just a little bit. These guys are held together by hydrogen bonds. Okay, if I could imagine the loops, what's holding these loops together? Hydrogen bonds. Finally, if I bend it one more time, again and again, I get a tertiary structure. It starts to take on a globular shape here. And if I combine this tertiary structure with another tertiary structure, and they combine, I get my quaternary structure, and this is a fully functional protein. Sometimes proteins can exist alone in the tertiary, but more likely than not, it's going to combine with another tertiary, become a quaternary, and then a fully functional protein. Hey guys, last thing, I think we've already talked about this before, but if you change the DNA sequence up here at all, that means you're going to change the amino acid sequence as well. And that could have profound consequences, just like the case of sickle cell blood cells. You can see it's normally CTC. If we change that to CAC, that one small change changes the amino acid from glutamic acid to valine, and that has severe consequences. If there's anything else left on this, this is a cool website if you get a chance to play with it. In fact, I think I'll put a link up to it on the lesson plans. And basically what it is go in here and just playing with how proteins fold back and forth. Now, it's not only playing, what you're doing is you're contributing to science. And if you're able to figure out a way to build a protein that nobody else has figured out before, it rewards you. And you get to make a discovery. So kind of fun to play with if you haven't already. Hey guys, let's visit the lesson plans really quick. For Tuesday, August 30th, guys, hopefully we got the review of this. I had originally planned on giving you a double helix coloring activity. And if you are done with the biologically important molecules, if you are done with properties of water, you're welcome to work with that. I'm going to call this one an optional activity, so I've crossed it out. However, it's been brought to my attention that students could use a little bit more time with both the biologically important molecules activity as well as the properties of water activity stations. Guys, let's give Tuesday as a day to work on these. And what I would ask, if you are a person who has finished both of these assignments and you feel pretty comfortable with the content, if you wouldn't mind helping out your classmates who might be struggling. And guys, again, that doesn't mean just opening your bill and showing them what you have for your answers, but actually going and looking at their bill, giving them ideas. I would say if you're struggling with a biological important molecules activity, make sure that you read the directions. I know they're long, they're tedious, and it's not fun to read, but it's going to give you some really important hints about which molecule belongs where. Now, I plan to be back on Thursday, and so if you have questions on either of these two activities, I'll plan to make myself available on Thursday, but we definitely plan to discuss this Friday as well. Guys, I heard nothing but good reports, so thank you for your good work. Know that if you have specific questions, please email me. I see there are some in my emails tonight that I need to go through and answer, and I'll try to get those before we get going here too long. So have a good day. Thanks.